Hi, I'm Olivia Williams, a biology PhD student in the Dameth Lab. Our lab focuses broadly on population genetics, but we also study the evolution of behavior as a consequence of selection. Today, I'll be discussing work from our lab on how the loser effect is influenced by protein synthesis. In life, organisms often compete with one another for resources. Loser effect is defined as the propensity to lose more resource contests after losing an initial round. Behaviors associated with loser effect, like fleeing or submissive gestures, are observed across animal groups. In vertebrates, one may see loser effect called social defeat stress. Social defeat stress is associated with PTSD and other human psychological disorders, some of which have a significant genetic component. Protein synthesis inhibitors like cyclohexamide, CHX, have been experimentally associated with memory impairment in mice and Drosophila. It's possible that memory consolidation, or the conversion of short to long-term memory, which is likely influenced by protein synthesis, is important for the behaviors associated with loser effect. Our model system for this research was the male broadhorn flower beetle, Nothoceros cornutus. Mating contests in this beetle have previously been demonstrated to lead to a loser effect, where the losing beetle flees the flying site and fails to engage in competition for up to 4 days. The goal of the study was to expand our understanding of the mechanisms of loser effect by testing whether protein synthesis is required for loser effect related behaviors in broadhorn flower beetles. Our hypothesis was that protein synthesis would eliminate loser behaviors. This experiment began with the rearing of naive male beetles from larvae. Once they reached adult eclosion, we fought the beetles to establish an initial group of losing individuals. Then we conducted a second round of fights. One round where losing beetles with or without CHX treatment faced an opponent that they had not seen before, and another round where beetles exposed to CHX faced their first round opponent. During these trials, we measured time to first retreat, or the time that it took for losers to give up on fighting, and time to loser effect shutdown, when the loser beetle stopped moving. Previous research in this system shows that bigger G. cornutus with bigger mandibular horns tend to win fights more often. This means that the effect of size could obscure the effect of CHX dose on our observations. Figure 1 from our results accounts for this variable by showing that weight difference between winning and losing beetles does not impact time to LE shutdown or time to first retreat, but CHX treatment does. Figure 2 demonstrates the two main findings of this study. First, we noted from our data that CHX treated losing G. cornutus delay both time to first retreat and transition into LE shutdown, as demonstrated by the higher median in the orange box and whisker plot. This suggests that ongoing protein synthesis is likely required for the typical loser effect presentation in G. cornutus. Second, the figure shows that CHX dose losers in familiar trials entered loser effect shutdown much more rapidly than their CHX dosed unfamiliar counterparts, but more slowly to loser competitors that were not dosed with CHX but still competed in unfamiliar trials. This led us to the idea that loser effect may involve independent components of both opponent recognition and memory consolidation. Further directions for this research may involve leveraging existing time series RNA-seq data to conduct RNAi or pharmaceutical evaluation of gene candidates controlling this response pathway in Nothoceros. This will help us not only better understand conserved molecular mechanisms of loser effect across animals, but clarify why only some stress responses lead to loser effect with a prolonged duration or specific effects on activity and health.